Bismillahirrahmanirrahim. Today I will continue my second video about the interesting process, NETosis, or the formation of neutrophil extracellular traps. And I hope that you had enjoyed the first video about NETosis and you understood it, so you can follow what comes in this video today. Today we will discuss in this video the role of NETosis or neutrophil extracellular traps in pathological conditions. I will continue to explain the information came in this nice and strong paper in biochemistry, NETosis, molecular mechanisms, role in physiology and pathology. As I said in the first video, NETosis is a program for the formation of neutrophil extracellular traps or nets, which consists of modified chromatin decorated with bactericidal proteins from granules and cytoplasm. And again, here is the diagram that I explained in details in the first video. To summarize, the process of NETosis depends on increasing the concentration of reactive oxygen species that came mainly, the main way is through activation of the net pH oxidase through activation by its enzyme protein kinase C. This enzyme will be activated through many ways. One of them is through mitochondria active oxygen species that come from increasing intracellular calcium that go inside the mitochondria and increase mitochondria active oxygen species and non-selective mitochondrial pores. And these reactive oxygen species will result in degranulation of the granules of neutrophils, and the elastases and other enzymes inside the granules will go to the nucleus and make histone modification and chromatin decondensation, and pores will be formed in the nuclear membrane and also in the plasma membrane, so that this big size chromatin will go outside and decorated with the contents of the granules from the neutrophilic cytoplasm. The first pathology that NETosis share or play a strong role in it is the thrombosis. And we have something called thromboinflammation, which I will discuss in details in another video. This thromboinflammation, the neutrophils by the process of NETosis play an important role in it, which involves the interaction with the endothelium and the platelets, and this NETosis apparently underlies thrombosis associated with excessive innate immune reaction in what's called also immunothrombosis. So immunothrombosis is the complex network between coagulation system and the innate immune system, and actually this thrombosis related to the innate immune system is just a protective reaction of the body against pathogens, facilitating its capture in the fibrin clot and preventing pathogen from going or spreading inside the whole body. NETosis can be activated also by microcrystals of cholesterol, and so NETosis can participate in the process of atherosclerosis. Actually, histones in the nets can cause toll-like receptor 4 dependent activation of macrophages and release of cytokines that will activate T helper 17, the main pro-inflammatory cell in the immune system. And this inflammatory process in atherosclerotic plaques can be one of the causes of thrombosis on top of it. Oncological disease also could be another cause of thrombosis, in which NETosis was found to be involved in many cases. Also in thrombosis, which is characteristic of severe viral infections, could be associated with severe NETosis, for example, in the pathogenesis of COVID-19 severity. NETosis also in the pancreatic ducts can be activated by microcrystals of calcium carbonate, which leads to blockage of pancreatic duct and pancreatitis. Also, crystals cause NETosis in the bile ducts in the gallbladder and could lead to the formation of gallstones. In this paper, in Frontiers Immunology, July 2018, its title, Neutrophil Extracellular Traps Formation and Aggregation Orchestrate Induction and Resolution of Sterile Crystal-Mediated Inflammation. Now I'm going to talk about another role of NETosis in pathology. NETosis in the role in pulmonary diseases. Actually, NETosis contributes to the protection against respiratory infections by increasing the viscosity of the mucus and the destruction of the pathogens. However, in many pathogens like Streptococcus pneumonia, Haemophilus influenza, have their defense mechanisms that include production of endonucleases, which break down the nets. While NETosis' original aim is the defense mechanism, in some situations NETosis contributes to the development of complications of the lungs in infectious disease. So it shares in diseases like acute respiratory distress syndrome, chronic obstructive pulmonary disease (COPD), and bronchial asthma. Researchers have found that acute lung injury and in acute respiratory distress syndrome of various etiologies, they are accompanied by excessive NETosis. Nets in these pathologies may be involved in the damage of alveolar epithelium and endothelium. In chronic obstructive lung disease, pulmonary disease, which is usually associated with smoking and air pollution and severe neutrophilic asthma, they are characterized by accumulation of nets in the sputum and the respiratory tract lavage. These nets is not only formed by traps from the neutrophil, it also formed from the death of eosinophils in a process similar to NETosis. Also, in severe infection with coronaviruses that are accompanied by viral pneumonia and progressing to acute respiratory distress syndrome and multiple organ failures, they found that there is elevated levels of neutrophils in the blood, and these levels indicating the severity of the disease and the poor prognosis. In severe cases of coronavirus, also they found increased level of NETosis markers, example cell-free DNA, myeloperoxidase, DNA complexes, and citrullinated histone. The marker of cell death, also lactate dehydrogenase, all these markers were elevated. Also, concentration of cell-free DNA was correlated with the content of neutrophils, and C-reactive protein marker of acute phase inflammation and D-dimer marker of thrombosis were found to be high and correlated to the severity of the disease. Other evidences of the role of NETosis in coronavirus infection, also they found that serum from patients induced NETosis in healthy donor blood. Also, one manifestation of severe COVID-19 is Kawasaki syndrome, which is accompanied by excessive NETosis. And another thing is that excessive NETosis is involved in the development of cytokine storm and thrombosis, which are main indicators of severe course of COVID-19. Another important role of NETosis in pathology is the role of NETosis in autoimmune diseases. Actually, many NET components, in particular double-strand DNA, granule proteins, and histones, can stimulate the production of antibodies and the development of autoimmune diseases like systemic lupus erythematosus. And they found also special form of granulocytes called low-density granulocytes that appear in the blood during these autoimmune diseases. This special form of granulocytes, low-density granulocytes, are characterized by a more production of inflammatory cytokines like interferons, tumor necrosis factor alpha, and also these low-density granulocytes are more involved in NETosis than the normal neutrophils. And their nets contain more autoantigen and oxidized mitochondrial DNA, which makes them more stronger as an immune stimulant. Also, one of the characters of low-density granulocyte is increased production of mitochondria-reactive oxygen species. Another example of the role of NETosis in autoimmune diseases is in rheumatoid arthritis, where the development of pathology is correlated with the accumulation of NETosis markers such as DNA myeloperoxidase complexes and antibodies against citrullinated histones. Also, nets engulfed by fibroblasts stimulate formation of antibodies against citrullinated histones in rheumatoid arthritis models. Similar observations indicate that NETosis is involved in other autoimmune diseases such as vasculitis associated with anti-neutrophil antibodies, antiphospholipid syndrome, multiple sclerosis, and psoriasis. Interestingly, on the other side, NETosis can play a role as an anti-inflammatory. For example, during gout attacks, NETosis induced by uric acid crystals is accompanied by release of lytic enzymes that break down pro-inflammatory cytokines in the foci of inflammation. So it's believed that NETosis in gout prevents the development of chronic disease. As regards the role of NETosis or neutrophil extracellular traps in skin diseases, I had this paper which was published in Biomedicine 2021 December, neutrophil extracellular traps in skin disease. In this paper, they mentioned that neutrophil-associated dermatologic diseases include many diseases like psoriasis, generalized pustular psoriasis, impetigo hematiformis, subcorneal pustular dermatosis, bioderma gangrenosum, hidradenitis suppurativa, Sweet syndrome, Behçet disease. So all these diseases are characterized by neutrophil accumulation. However, the clinical presentation is different between them, and also the degree and the layer of neutrophil infiltration varies between groups, and also the presence of netotic neutrophils is not the same in all of them. So we don't have evidence of netotic neutrophils in pustular psoriasis, like generalized pustular psoriasis and AGIP, but apparent cutaneous neutrophilia is clearly observed in this disease. As we know, myeloperoxidase mutations that present in some cases of generalized pustular psoriasis will lead to impaired neutrophil clearance, which could explain why there is an accumulation of neutrophils in these diseases. So that this accumulation is a consequence of dysregulated clearance of neutrophils and subsequent neutrophil accumulation. On the other hand, the presence of netotic neutrophils is evident in other diseases like psoriasis, in which netotic neutrophils amplify inflammation spirals. Also in Stevens-Johnson syndrome and TEN, they promote keratinocytes necroptosis. And in Bioderma gangrenosum and hidradenitis suppurativa, they can damage the dermis and the subcutaneous tissue, resulting in the formation of ulcers or scars. So while we don't have such evident NETosis in generalized pustular psoriasis, but we have it in other examples like psoriasis and Bioderma gangrenosum and Stevens-Johnson syndrome and TEN and hidradenitis suppurativa. In Behçet disease and Sweet syndrome, NETosis could be responsible for damaging blood vessels or the dermis, resulting in the formation of tender and indurated erythema. And as we know, in Behçet disease, there is increased incidence of venous thrombosis, which could be also related to the process of NETosis. And as regard the therapeutic trials to target NETosis in diseases, I found this very recent paper published in MedCom in September 2022, which means the last month, and it's titled Molecular Mechanism and Therapeutic Target of NETosis in Diseases. This diagram from the paper I just mentioned in the previous slide, which illustrate our trials to make therapy for NETosis. Like we have here calcineurin inhibitors like cyclosporin A, which could inhibit increasing intracellular calcium, which will lead to increase in mitochondria-active oxygen species and increase activation of NADPH oxidase enzyme. Also, we have PAD inhibitors. We know that PAD4 plays a central role in NETosis because PAD4 is responsible for making a process called histone citrullation, which changes histone residue from arginine to citrulline and changing its charge so that the chromatin can do decondensation. So using PAD4 inhibitors like C1-amidine hydroxychloroquine is under trial also. We have also toll-like receptor inhibitors like hydroxychloroquine or dexamethasone. These will act on toll-like receptor as a receptor that stimulates NETosis. We have also the diphenylene iodinium chloride, which work on inhibiting NADPH oxidase enzyme, which is called NOx also. And also we have reactive oxygen species scavengers to decrease the production or the availability of reactive oxygen species that will make degranulation of neutrophil granules and release of their enzymes like neutrophilic elastase that will go to the nucleus and start the process of NETosis. We have also antibiotics, which could act as in some way in the process of preventing the process of NETosis, like chloramphenicol, azithromycin, and gentamicin. We have also other medicines or other items that work by other ways, like vitamin D, 13 series, T series, resolvine, and tetrahydro isoquinolones. And we have also one important item is recombinant human DNase, which will destruct the net and trial to destructing the net itself and removing it. Again, this table is showing the different therapeutic trials to manage NETosis and in what stage of drug development they are. We have toll-like receptor inhibitors like dexamethasone in phase 4. Their target is toll-like receptor 2 and 4. And we have calcineurin inhibitors like cyclosporin A, which is in phase 2, that will act on calcineurin. And we have reactive oxygen species scavengers. We have different chemicals here in phase 2, phase 3, preclinical. Their target is reactive oxygen species. We have PAD inhibitors, as I said, like C1 amidine. It's a preclinical. We have DNase, which will act on the net, recombinant human DNase. In phase 1, this will act on net-derived DNA. And we have anti-inflammatory and immunomodulatory antibiotics like azithromycin, chloramphenicol, gentamicin. These are in phase 3, azithromycin, chloramphenicol preclinical, and gentamicin phase 4. And their target is cytokines. Other items, we have vitamin D, which is in phase 2, and 13 series resolvine, it's a preclinical, and macrophages, and others also will tetrahydro isoquinoline, so will work in preclinical and neutrophils. So to conclude, excessive NETosis has been suggested to play an important and central role in the pathogenesis of many infectious, inflammatory, and autoimmune diseases. So the development of new drugs that target NETosis and preventing it, including mitochondrial-targeted antioxidants, appears to be a promising area of pharmacological research. Lastly, I hope that you enjoyed the video and you understood the nice concept or the process of NETosis and its important role in many pathological conditions. And before ending, I want to remind you again to subscribe to the channel and to activate the alarming bell if you like to get any new videos from this channel. Thank you.